afternoon or good evening. Good morning, good afternoon, or good evening, depending on where you're joining us from. I'm Bruce Campbell. Welcome to the PNOC Immunotherapy webinar. This is our second webinar for 2022 and our first non-tumor type specific webinar discussing the promising and exciting field of immunotherapy with applications covering many pediatric brain tumors.
And I'm Alan Campbell, and together we helped co-found PNOC Foundation in 2013, about 3 years after our son was diagnosed with his brain tumor. PNOC Foundation is the public nonprofit charity really designed to help create fundraising, awareness, and support for the urgent work of these PNOC doctors, scientists, clinicians, and specialists.
Like many of you, I was excited and encouraged by research published in the New England Journal of Medicine last week, coming out of Providence Cancer Institute, related to an adult patient with advanced pancreatic cancer that has spread to the lungs after several treatments and has shown a remarkable response to CAR T cell therapy. Today, our panelists will talk about the immune system in children with a CNS tumor and the promising developments of immunotherapy in clinical trials for high-grade tumors, including DMG, DIPG, medulloblastoma, ATRT, ependymoma, and other tumor types.
Yeah, today, really the purpose is information sharing. And as Bruce had just said too, we have, unlike our previous webinars, a wide range of viewers from families with different tumor types. Some families are just being newly diagnosed, and some are really battling this disease for some time. So we really do want to hear from all of you. So please put your questions in the Q&A, and we'll be sure to answer all of those.
For those of you joining us for the first time, the Pacific Pediatric Neuro-oncology Consortium, or PNOC, is a leading global pediatric cancer consortium with over 35 study sites in the US, Europe, Australia, Israel, and India, striving to improve outcomes through a unique collaborative approach, translating the latest findings in brain tumor biology into clinical trials and more effective treatments. A key mechanism to support the translational research is through the unique PNOC working groups, which include investigators from around the world with specific tumor type or specialty expertise. Through scientific collaboration and consensus, the most promising clinical trials are developed. Advocates and philanthropists are also invited to lend their perspective and partnership. It's a great honor to introduce today's powerhouse panel.
Yes, we are very lucky. Today we've got Dr. Sabine Mueller, PNOC co-founder and project leader. She's also an assistant professor of clinical neurology at UCSF. We have Dr. Jessica Foster, pediatric neuro-oncologist, Children's Hospital of Philadelphia, co-leader for PNOC Immunotherapy Working Group. We have Dr. Nicholas Vitanza, assistant professor, University of Washington School of Medicine, pediatric neuro-oncologist, Seattle Children's Hospital. Dr. Jasper van der Lucht, pediatric oncologist, lead of the neuro-oncology subdivision of the Trial and Data Center, Princess Maxima Center, PNOC lead for immunotherapy, and Dr. Hideo Okada, professor of neurosurgery at UCSF, member of the Parker Institute for Cancer Immunotherapy, associate editor of the Neuro-oncology Journal. Welcome to all of you. Thanks for joining us.
Thanks, Alan. Let's turn it over to our panelists to give us an overview of immunotherapy and its application to pediatric brain cancers.
Thanks so much. I'm just going to share the screen. We have a few slides to share. So thank you again to everyone who's joined. And again, we welcome all your questions. Please feel free to ask us anything along the way. And we'll just start with a brief introduction, starting with the basics, which is an overview of the immune system. And so the immune system is divided into what's called innate immunity and adaptive immunity. And innate immunity is sort of things that are already in place all the time to try to help prevent infections and other bad things from entering your body, as opposed to adaptive immunity is a little bit more specialized. It's where things get fine-tuned and really go after particular targets. And so in innate immunity, that's where we have things like our skin, our GI tract, other things that are just barriers. There are other specialized cells that are in that category, including ones that kind of eat up bad pathogens. Those are called phagocytes. Those are here, and you can see those are something called dendritic cells and macrophages. There's other components as well, little proteins called complement that also play a role, and a particular type of white blood cells called NK cells, that stands for natural killer cells. So all of those are always there, sitting ready, waiting to act, to help prevent any infections and other things from coming into your body. Whereas the adaptive immunity really sets into gear once something has made its way in. You can see a microbe here kind of getting past the epithelial barrier. Once it gets past there and the innate immunity has been revved up, that's when it's these some of these cells, the dendritic cells, will actually partner with other lymphocytes, specifically your T lymphocytes, you can see down here, to really tell them to get active, to multiply, and to go out and to kill whatever it is the target they're going after. And your B cells also have these antibodies on the surface that recognize the pathogen and then start making many, many more antibodies to go after whatever the bad thing is that's entered your body. And so this is just a brief overview of the immune system that I think is important to start there and start with what we're talking about before taking it to the next level, which is then immunotherapy. So immunotherapy is this broad umbrella term we use anytime we're using the immune system as a way to try to target, in our case, cancer cells. And so you can do that through so many different mechanisms, and you can see them listed here. So when we do things like using a vaccine or using something called an oncolytic virus, it's truly a virus, we're really trying to imitate that microbe that's entering the body, and really from soup to nuts trying to engage the immune system all the way across with those mechanisms. So it's really trying to essentially mimic this foreign pathogen that's coming into the body. We can do additional things to try to manipulate some of those cells that are the ones that eat the, whatever the foreign material is. Sometimes those cells can sit in the tumor and actually send a negative signal out to try to calm down the immune system. And so one of the ways that immunotherapy is actively working is looking at sort of inhibiting those cells and turning off their negative signals, in particular macrophages and microglia. Those are two of those cells that tend to sit there in the tumor tissue and send a negative a negative signal. Oops, excuse me. The NK cells are also an area that people are actively looking at. Again, you can take those NK cells and expand them up, and there's work actually within PNOC looking at that and using those as potential killer cells against the tumor cells. And you can actually take NK cells and manipulate them to have proteins on the surface, which we'll talk about a little later, called a CAR, or a chimeric antigen receptor. And so there are some groups as well doing research there within those NK cells. And then over here on the adaptive immunity side, again, immunotherapy can include other different types and actually a lot of immunotherapy is focused on this side of the screen there's antibodies. Again, you could just take antibodies against a particular target and infuse those. You can also give antibodies that are inhibitory for something that trying to block a protein. Antibody drug conjugates, where this is where you have an antibody that has a drug actually connected to it. So the antibody will go stick to whatever it is, your tumor cell is on your tumor cell. And then actually it will take that antibody inside it, and the drug will then act on the tumor cell. That's how those antibody drug conjugates work. And then over here for the T cells, again, the T cells are really very targeted killers in the immune system. And so a lot of energy is focused on them and trying to make them maximally effective against tumors. And so using checkpoint inhibitors, those are actually, they're an antibody, as I mentioned before, that's really inhibiting a particular signal. It's really taking off the brakes on the T cells. You can also just take T cells and expand them up, regular old T cells, and try to make a whole lot of them. You can try to specifically pick out T cells that will go after a particular tumor target and expand those out. Those are the expanded T cells, or cytotoxic T lymphocytes, or CTLs. And then there's two other mechanisms that you can actually engineer the T cells, and we can talk about those in a little bit more depth in a minute. Those are T cell receptors, or engineered T cell receptors, and again the chimeric antigen receptor, or CARs, that we talked about. But this is really, again, just a broad overview of the immune system and then immunotherapy and how it can play, we can use it in many different ways, and there's many different facets of the immune system that we can use. And I'll turn it over to Jasper now to talk about the next slide.
Yeah, so thank you very much, and I'm honored to be here as the only European physician here. So actually, this is a kind of cartoon of what is happening with all the different immune cells Jessica just talked about, and I don't go into detail for each individual cartoon. This is are the tumor cells. It looks a bit like the brain, but it's supposed to be the tumor. And there you see like all kind of cells who have specific functions or should have specific functions to attack the brain. But what happens, it's brain tumors or solid tumors, brain tumors in particular, there are quite some immune cells that protect the tumor. So here you see with this police hat protecting the tumor, and there's a T cell that is protecting the tumor from being attacked. And here what you see, there's a kind of engagement of the tumor with this normally active T cells, and therefore he gets exhausted and is not recognizing the tumor as being wrong. So there are all kind of mechanisms of this tumor. Here also there's some interleukin 10 is produced by the tumor to avoid inflammation. So there are all kind of mechanisms that protect the tumor for getting attacked, and it's very important to get to know what's actually happening, because then you may understand why immunotherapy is working or not. And I think there are very promising results in adults, and starts to get also into the pediatric field. Brain tumors are still very hard for immunotherapy to reach and to be effective. So we need to understand what's happening. And so here it's also a bit of a cartoon where like all walls around the tumor, and all these cells are trying to enter the tumor, but there's like only very few chances to get towards the tumor, and once they're there, they get very easily arrested by the tumor. So we need to break this wall down basically and see how we can get the tumor. This is maybe a bit too detailed. There are all reasons or all challenges we need to take into account once we are actually conducting treatment with immunotherapy, and why it's challenging to get brain tumors vulnerable for immunotherapy. So we call it like cold tumors, immune privileged. I mean, their immune system is definitely different than the rest of the body. There are definitely also interaction with the immune system outside the brain. So we need to get to know what is actually happening, and then we can improve our immunotherapy. So therefore, for the next slide, the next slide. So now, for example, we perform a program in the Netherlands for getting a lot of material from our patients who are having a brain tumors, just to get better information on the immune system. So not only the tumor itself, but also the CUSA material, and that's the material that is the CUSA. This like a machine, is like a vacuum cleaner that is very used in the operation theater to get pieces of the tumor to not make too much damage. And all these kind of small pieces of tumor we can also use for investigation what's happening with the immune system, of course, the blood, the cerebrospinal fluid, and even if the patient allows us, we can look into the bone marrow, because that's a very important source of the immune system. And even nowadays, microbiome, the Thesis, is also a very interesting place to look at for immunotherapy. And this, I don't go into what we all are performing, but these are all different methods how we profile the immune system. So just an example of an apartment we look into, that are the Thesis. We ask the patients to collect it. And then we look into the microbiome. The microbiome is basically the compositions of bacteria that are in the microbiome. So everybody has a more or less different microbiome, but there are also a lot of similarities. And we find out the last decades that microbiome plays a very important role in all kinds of diseases. So autoimmune diseases, also graft versus host disease. So we know more and more that there's a strong interplay between the microbiome and all kinds of diseases. And we need to find out, here is this arrow here, if this also holds true for brain tumors, in particular treatments for brain tumors. Why is it important for oncological treatments? Now, for recently, there has been, this is a CD19 CAR, so for leukemia, they have looked into the microbiome and also looked into antibiotic use. And what they have found is that patients who were using antibiotics 4 weeks before starting the CARs, they performed much worse than the patients who did not use the antibiotic before 4 weeks before they were getting the CARs. So you see the progression-free survival in gray is the patients who did not get antibiotics. The other one is who did get the antibiotics. So quite a difference, and the same is for overall survival. The same is also true that the microbiome can predict if you have more or less toxicity. So these are all kind of different species. You have the names are endless of this bacteria. You see that the red ones, you see they predict for more toxicity, and the blue ones predict for less toxicity. So it's very interesting to look into this microbiome, even find out with a certain type of immune therapy, so checkpoint inhibition, that's the one who's been used quite broadly for melanomas, lung cancer, etc. The microbiome is also predicting the response, and they now have the first trials that they do fecal transplantation. So they give another microbiome to a patient, and then they see after they have progression, they do transplantation to give the same immunotherapy, and that quite a part of those patients who respond again. So this is very exciting. That needs to be developed much further, but that's just to let you know there's a lot of happening in the microbiome. Next slide. Yeah, so that's what I just said. Next slide. Yeah, so very briefly, we also look in the blood of the patients. So and everybody just heard that their Tcells are very important, and you have CD4 and CD8 cells, and they looked into adult patients, and they saw at diagnosis of a glioblastoma, the patient had very low blood count of CD4 even before they start treatment. So you see here they have very low amount of CD4 compared to the control group, and the same is true for CD8. And actually you need those cells to protect yourself, but also maybe they can be effective against the cancer. So we need to see why this is happening. And we also looked into in our patients, so at baseline, and also here you see the lower threshold is the dotted line. And there are quite some patients below that line at diagnosis, and the same is true for CD8. So a lot to explore, and understanding the immune system in all different compartments may bring us further in developing new immunotherapy, and also to monitor what's happening once you start immunotherapy.
We can keep going unless anybody wants us to stop, but we can keep going to discuss a little bit more in detail some of the other types of immunotherapy.
Yeah, go right ahead, Jess. Thank you.
And I don't know if Dr. Okada wants to tackle the TCRs. He may be in transition here, so I'll talk about them as well as the CAR T cells real quickly. But Dr. Okada, please interrupt me at any point. So TCR stands for T cell receptor, and so as we mentioned, you know, the way that T cells work is they meet up with antigen, what's called antigen presenting cells. These are other cells that we talked about that have swallowed something from, you know, either the virus or whatever bacteria it is that is the pathogen. They then kind of connect here with the receptor and the antigen presenting cell, or the cell that has swallowed, again, the bad thing, tells the T lymphocyte, yes, your receptor is a match against this particular target. Go ahead and grow. So they grow. And again, now you have a big pool of these T cells that have a particular target that can then go after, again, cells that have whatever it is, the target you're going after. So we're going to zoom in here on this little interaction between the T cells and in this case, a tumor cell. But you can see, so this is just a very zoomed in picture, kind of turned sideways, where the tumor cells on the top and the T cells on the bottom. And so this T cell receptor is made up of a lot of different proteins. It's actually a really complex structure, but it's this beautifully intricate receptor that will only match a very particular target. And once it does, it sends these signals, and it has to have a couple other proteins as well that line up just the right way. And when the stars all align, it's what is the beautiful thing about T cells that makes them these wonderful targeted killers. And so this is, again, the endogenous, or the T cell's own receptor. It comes from them. This is how they're naturally made. On the right is an example of where you can actually make one in a lab. You can actually, you know, create the different protein structures that go together and then will form, again, with some of these extra proteins on the side that are naturally in the T cell, to come together to form a T cell receptor that looks really pretty darn similar to the one that's there already on the cell. And so again, if you have engineered it right in the lab and you figured out exactly where you want it, what target you want it to go after, again, you'll have something that's being presented by the tumor cell that will then activate the T cell naturally, and it will go on and kill. So that is the T cell receptor. The switching gears very quickly, just again for comparison's sake, that's in contrast to CAR T cells. So again, CAR T cells stands for chimeric antigen receptor. And again, just to go over it for those who aren't familiar, the way this works is that T cells are actually collected from a patient. So it starts with a process where you just take all the blood out, and actually specifically the white blood cells, and you sort out for the T cells, and you get them to start dividing artificially in the lab. You can do that through a couple of different ways. And then in the lab, we put something different, not the T cell receptor, but a chimeric antigen receptor, and we'll see that on the next slide, onto the top of the cell, and then you give it back to the patient. And so the way that CAR T cells work is, this is, again, the zoomed in picture of the CAR. So the top half of it is essentially the top half of an antibody. So it's essentially a protein that binds the protein target, or whatever your target is on the tumor. It doesn't have to be a protein. The bottom half of it is this artificial Frankenstein molecule that signals the T cell to kill. And so that's again, this is the structure that's made in the lab. A little bit different than the TCR, but does the same job, where it signals to the T cell to kill. And so that gets put on the top of the T cell. Again, these T cells go through their quality control, get given back to a patient, then they travel through the body. Where they go, they find their tumor cell, and engage with their target, and that's what stimulates the T cell to do its killing. And so that's just a general overview of the TCR, the T cell receptors, and the CAR T cells. And then just a couple of translational considerations, which is true really for, it's written here for CAR T cells in the central nervous system, although probably true for almost all immunotherapy, to be honest. We have to think about different things as we think about bringing this into patients. There are barriers, and this is both physical barriers, which gets a little bit into the delivery. There's some protected space, as Jasper mentioned early on, that we think about the central nervous system as being somewhat protected from the body. But there's also barriers within the tumor where, again, they're sending these negative signals and other things that might be keeping the immunotherapy away. So there's a lot of different barriers we have to keep in mind. The delivery question is one that I think comes up a lot, where we think about where is the best place to put these immunotherapies, or in my world and in Nick's world, we often think about it in CAR T cells. Do we put it directly into the fluid around the brain? Do we put it directly into the tumor? Do we give it to into the vein? So those are some of the considerations and questions that I think are still outstanding in the field. Picking the right target. Again, I think this is one of the biggest and hardest things that a lot of folks are actively working on. We're trying to figure out what is there on the tumor that we can go after, and what will work to actually engage, and what do we have a good binder to bind and go after the tumor. And along those same lines, ideally, we want a target that's on the tumor, but not on normal tissue, because then that gets into toxicity questions, because there can be issues of whatever you're giving that might go after normal tissue. In addition, there's additional toxicity that comes just from having your immune system revved up alone, having your immune system revved up in the brain specifically. So there's a lot of different pieces we have to consider as we're working on this, and putting this all together. I believe the next slide. So then we just have one quick slide, and Jasper, if you want to take over here about oncolytic virus, we'll just talk about one other, again, form of immunotherapy, and then take a pause.
Yeah, thanks. Yeah, so very briefly indeed, this is also another modality of immunotherapy, and it's called oncolytical thyrotherapy, and viruses tend to infect fragile cells, and cancer cells are in general fragile. And moreover, the oncolytic virus can be manipulated that they only infect cancer cells. And the idea is that you locally inject the virus into the tumor, and this virus is actually infecting all tumor cells. And by replicating into the tumor cells, it replicates, and therefore it lysis of the cell, it dies. And after it dies, you get all kind of tumor-associated antigens, all kind of inflammation happening, and this inflammation can again activate your immune system, and this immune system then starts to recognize the other tumor cells as well. And then in our most ideal situation, you get rid of other tumor cells as well, because it's recognized by your own immune system. So there have been some trials also in the glioblastomas with some moderate positive results. The good thing about the oncolytical virus is that it changes the microenvironment we just discussed. It can also be something that you need to combine with another a way of immune therapy. But locally delivery is very attractive for this type of therapy, and 2 hits you hopefully get when you inject this virus.
Excellent overview. I've got to say, I've seen a number of presentations on immunotherapy, and by far, actually, that is the best overview, at least for me, that I've actually seen. Really, I loved the diagrams, the visual aids really helped to understand the overview of actually how it works and the different options to use immunotherapy. Now, it's interesting, we don't have a lot of questions today. Usually we're inundated with questions, and I think that reflects just how new and novel a lot of this work is to pediatric brain cancers. That there's not necessarily a lot of knowledge or a lot of questions because of its newness. What I'd love to understand is a little bit more specifically about, how PNOC is using immunotherapy, both currently and potentially what's on the drawing board in terms of, where PNOC goes next with the use of immunotherapy.
Yes, I think there are a few questions, but I will answer that question first, Bruce, maybe. And so where PNOC is going, so we have invested and are currently conducting trials with oncolytic viruses that Jasper just mentioned. And we are actually working in the laboratory with including Dr. Okada and some other experts around the country to really think about what would be the best combination strategies. We also have currently trials open that are looking at the side that Dr. Foster was showing on the right-hand sort of checkpoint inhibition, but also with the macrophages. We just started a new trial using an antibody that basically prevents the sort of don't eat me signal, and we are in active investigations how to further expand this. And then we are starting a new trial with a T-cell receptor approach that also Dr. Foster just beautifully highlighted within the diffuse midline glioma setting against the H3.3 mutation. And this is based on work from Dr. Okada. And then we have a checkpoint inhibitor trial trying to look at the combination of different radiation regimens. And then we have a very exciting trial. This is led out of work by our team in Florida, looking at an RNA vaccine against high-grade glioma. So there's a lot of activity really covering sort of a spectrum that was sort of presented really from a vaccine-based approach. And we have an ongoing trial, though, that completed accrual using a specific peptide vaccine, also in the diffuse midline glioma setting, and using it in combination with a checkpoint inhibitor, from oncolytic viruses to now going into this more area of T-cell receptor. And I think one question that frequently comes up from parents and families, and I will address this to Dr. Vitanza, who maybe really has a lot, maybe the most experience currently with treating patients with brain tumors on these novel CAR T-cell therapies. So how do you go about investigating these? One question was, if you are assessing these in models that maybe are not immune competent, how sure can you be about your results? But then also, if you advise families right now on these different strategies and how you do target selection, if you could broadly comment on that, that would be great.
Sure. Thank you, Sabine, and thank you everybody for giving me the opportunity to speak here. I'm actually at an immunotherapy symposium in Milwaukee while on an immunotherapy call here. And happy to be able to join and talk about some of this work. For those of you that don't know, I'm at Seattle Children's and oversee 3 CAR T-cell trials for children with a variety of brain tumors, in fact, any recurrent brain tumor or DIPG. And these are trials in which we've now enrolled 55 kids and given 245 intracranial CAR T-cell infusions, including 83 infusions just to children with DIPG. To answer the question about xenograft DIPG models. So for those of you that don't know, the way that we study tumors in the lab is a variety of ways. One, you can take a normal piece of tissue and genetically engineer it to become a tumor, or you can take a patient's tumor and bring it to the lab and try to get it to grow so it can be studied. Those are the 2 most common ways. Michel Mange really helped pioneer having very generously donated tissue from children at the time of their death at autopsy brought to the lab to develop models. And since then, multiple of our labs have also been able to get biopsy samples from children's initial diagnosis to grow tumors in the lab that grow either in a dish or in a mouse. And then essentially it's a mouse with a human DIPG. But the way that we, if I lived in New York City for a long time, if you just take a rat that's running around the street, try to put in a pediatric brain tumor, it's not going to grow because of the mouse's immune system. So what the question here is asking is, do most of our models not have immune systems? And the answer is yes. So I would say probably on the order of 98% of pediatric brain tumor research that happens in the lab is in animals that don't have immune systems. And that's how we get a human tumor to grow, because if it had a normal immune system, it would reject the tumor. What does this mean from a practical sense? Well, Jess and Jesper did a really great job of describing the immune system. If we just focus on T cells, type of white blood cell, they wouldn't really be present at normal levels in a mouse without an immune system. And so you put in the brain tumor, the mouse has a growing, let's just say DIPG tumor, and then you give it CAR T cells, you are now putting an immune system in a place where there wasn't one before, and it has the potential to make your results look artificially good. So the way that I think of our studies is, what we're doing is making sure that the CAR T cell has biologic activity. So we can take CAR T cells and put them in a dish with tumor cells and show, so there's not an immune system that we have to worry about in the dish. And you can see what's the percentage of lysis of the cells based on the CAR T cells. Are we getting other immune cytokine production showing that immune response is happening? And then when we move to animal studies, we're looking at safety, and we're also looking to see if you still have that biologic effect. So people like Stanford's group have published on GD2, and we've published on HER2 and coming up B7H3, and multiple other groups have published on these things, including Baylor and City of Hope, that CAR T cells can be curative for these mice in the lab. And that is obviously has the bias of these mice not having immune systems. What we, to overcome that, when we open our trials, we take that into account. So we give one dose to a mouse, we cure the tumor. We unfortunately don't have the expectation that would happen in a child. And that's why most of our studies are built where children get repeated dosing of CAR T cells. So we try to admit the limitations that we have in the lab and build them into the studies. The reason that we don't study animals with normal immune systems is that they exist, but as opposed to regular, just throwing, let's say it costs $10 to get a regular mouse that you're studying. It might cost like between $250 to $1,000 to make a mouse with a normal immune system. So the cost of your laboratory studies goes up dramatically by doing that. And it's also very hard to even feasibly construct. So there's not even very good models to use, and it can take years and years to develop a good model that has a normal immune system. So I talked way too long about that answer.
I have one extra thing too.
Yeah.
There are some differences. So I am actually actively trying to do an immune competent diffuse midline glioma model and CAR T cells. And one of the other challenges is mouse T cells are a little bit different than human T cells. And so you do have to make some adjustments in the CAR. And so it's not just a one-to-one where you can flip in the CAR from the human and do it to a mouse. You have to, there's some of those little extra side proteins have to be adjusted. Some of the stimulatory domains on the inside have to get changed. So it can be done, but again, I think Nick and has mentioned most of the big challenges and hurdles, as well as this other extra piece that unfortunately mice are just not tiny humans.
There was one other question from the audience, which basically is asking about checkpoint inhibition for brain tumors, and has that been really effective? And I can tell a little bit our experience. I would say as single agents, so far it hasn't been shown great efficacy, but there are exciting studies. And one of the studies was published now a few years ago, where I think we just don't fully understand yet how and when to best give these checkpoint inhibitors. There was a study done in recurrent glioblastoma, where when patients received one dose of a checkpoint inhibitor prior of an additional surgery for their recurrence, that these patients had a really significant improved survival. And the thought is that through the surgery, we are creating a massive immune response. And if we prepare the body with a checkpoint inhibitor for that could really stimulate sort of the immune system really for targeting the tumor cells. So we currently are also having a trial testing the strategy in the pediatric patients with recurrent high-grade gliomas open in PNOC. And we are looking right now in the laboratory, as I mentioned before, how we can best combine the checkpoint inhibitors. But I think as a single agent, and I would love to hear what other panelists have to say, it hasn't been the big breakthrough as we have seen, for other cancer types like melanoma.
So I think the only exception is, of course, the group who has the congenital mismatch repair syndrome. That's a cancer predisposition syndrome. So that means that you're having genes that need to repair your genetic mistakes. There is something wrong. So you have all kinds of mutations and you're very prone to develop cancer. And if you get cancer, these cancers are very, as we call it, hyper mutated. So you don't have like 1 or 2 mutations, which you have in pediatric cancer. There are very low mutational burden, a very high mutational burden. And therefore, we have all kind of more antigens presenting, and therefore you get more activated immune system. And then with this group of patients, you see if you use checkpoint inhibition, it does work, not in all, but for, and the more mutations you have, the more likely it is that it's working, not endlessly, but it has very promising results. And as a concept is of interest, of course, that if you change this environment or get this tumors into from cold into hot, something is happening, but otherwise we don't see any positive results from checkpoint inhibition.
Thank you. Great answers and so interesting. I've just been notified that we actually do have a patient family that has taken some time to join us today, and I really wanted to give them a warm welcome. Ashley and Nate. So nice to meet you. It looks like we have Gavin joining us too. Thank you so much for joining us this morning, making some time. I really want to hear, we want to hear, you're getting a nice warm hello from Nick. We'd love to hear a little bit about your journey and your story about your adorable son there.
Morning, everyone.
Morning.
And, hey, so I'm Nate. This is my wife, Ashley. And as you might have seen, that was Gavin, who's doing pretty good today. Yesterday was his 19th CAR T infusion. But yeah, I guess stop me if we get too detailed, but we'll start, I guess, from the beginning. So it was February 18th of last year was the day he was diagnosed with DIPG. Leading up to that, I think it was late December, early January, he kind of presented with eye movements that were odd to us, and we initially thought it was strabismus of the eye, but not giving too many details. We finally got in for an MRI, and that day, February 18th, was when we found out the news. Started radiation, I think it was the 25th of February. So did 30 days of radiation, and 30 days, and it was a little bit tough going through that, of course. Having such a young child, because he was 2 at the time, having been a month removed from his 2nd birthday. But as you can maybe hear in the background, not stopping him right now. But yeah, he went through radiation. I think he did some, was it Avastin?
Yeah,
A chemo drug during the
Avastin,
radiation.
and temozolomide.
So we had like a maintenance period after the radiation where we did some chemo drugs, which was tough for him because we had to do liquid at the beginning. Thankfully for us, yeah, and it was not fun. He quickly started to hate all sorts of liquid medications, but thankfully he learned how to swallow pills. So he started to swallow pills by, I think, June of last year. And so yeah, we went through the maintenance period and started our CAR T trial in September, late September, I believe it was. And it was the first few were tough because of his reactions at the time. That night after the infusion, the next day, he was very nauseated and kind of like headaches and stuff. But it was very hard to, I guess, communicate with him because he was being a 3-year-old, not exactly telling you everything you need to know. So that was a struggle to figure out exactly what his issues were. But probably by the sixth infusion or so, things started to kind of even out. You know, his reactions and symptoms after an infusion kind of became more predictable. And it was before that the day after was very much a rest day. He would not be up and around like this. And so now we're on number 19 and he's flying around like crazy the day after. But yeah, it kind of, what we normally see from him now with our infusions is maybe he gets a little nauseated, a little tired. But thankfully for us, that's a bit about it. I know a lot of DIPG families and patients can't say the same thing. So we're very thankful and lucky for that and the time we have. And also very thankful that, you know, we live so close to Children's. We're actually here about 40 minutes drive from Seattle Children's. So it's right in our backyard, and very thankful to have them so close. I think that's about it as far as the very broad overview. So I don't know if there's any like specific questions or anything that you guys would like to know from us.
Well, thank you for sharing that, Nate, and please pass that along to Ashley too. You know what I have to say is so heartwarming in seeing that energy in your son, Gavin. I mean, that's a real 3-year-old running up and down the hallway and squealing, and that's amazing. And I know Nick was your treating physician, and I'm not sure if he wants to add something more to that thought about some of the work that he's done. But, and the last thing I want to say is thank you too for offering to answer any questions for other patient families. It's very kind of you. And I'm sure that even if people don't offer them here, we will usually get them after the event. So thank you. And I know that they feel, you know, really comforted by the sense of community. So, and certainly by the hope of Gavin's outcome. Thank you.
Hey, Nate and Ashley. Hi, Gavin. Thank you guys so much for taking the time to do this. I don't think I have too much more to add. I think, you know, the trials that, when we're looking at immunotherapy, I would say that there's different ways to think about it. One of the advantages of CAR T cells is that you don't necessarily have to think of tumors in different disease groups, because a lot of different tumors have the same things expressed on their surface. So like of our 3 trials, we enroll multiple disease groups on all of them, but it was only our later trial that enrolled children with DIPG, as we got some safety experience first. And Gavin's been on our 2nd dose regimen, so now getting our 2nd level dose for children with DIPG. And so he gets them on an every other week basis. And there's multiple ways to give these therapies. Some work best IV, some work best intracranial. For some families who aren't experienced, what you can't notice on Gavin is he essentially has a port for his head. So under his scalp, he has a little plastic button, and then we use a butterfly needle, the same thing you would use to put an IV in the hand. And he comes to clinic and he's awake, and we numb the area and then put in the needle, and then he gets, when I say we give the infusions, what I mean is our nurse practitioners, I should say. I used to do our infusions, and then luckily we transitioned to the nurse practitioners doing them. But Gavin stays awake and he gets his infusion just over a couple of minutes. And what Nate and Ashley talked about with the nausea and vomiting and headaches is the most common side effects we see that occur the night of the infusion and then self-resolve. And interestingly for Gavin, he's had less and less side effects as he's gone along, although not everyone has the same experience. But I think that their discussion on chemotherapy being difficult is an important point, because a lot of times we focus on immunotherapy being like a next generation technology. And I will say, actually, immunotherapy has been around for a long time, and CAR T cells have been around for 30 years, and they're just being optimized and brought to these patients now. But we're really standing on the shoulders of giants with all of these things. But the other advantage that we hope is that they'll be better tolerated. So, you know, children like Gavin deserve to spend as much time out of the hospital as possible. So now he comes in every 2 weeks for his infusion and some other times for check-ins. But the goal is to not spend nights in the hospital and offer the most benefit as possible while letting kids be kids. And then obviously it takes amazing parents to be able to manage 3-year-olds at all, plus 3-year-olds on a clinical trial, plus 3-year-olds who are getting intracranial immunotherapy. So obviously that's not just the kids who are heroic. So thank you guys for making time.
So there were a couple questions, to expand on this. So one for Jessica directly, will there be CAR T cell trials at CHOP? And then the question is the limitation of the age. Will some of these CAR T cells also be opening up to older patients, adults? I think that was maybe a question more directed to Nick. Any comments?
Yeah, I'm happy to answer the CHOP question. Yes, we are actively working on opening protocols here. I am very, very happy that we, within the last couple of years, recruited Dr. Cassie Klein here to CHOP, who is just phenomenal at, phase 1 trial protocol development. And so together, she and I are working on opening trials here specifically, but also partnering with other sites and companies to hopefully also open some of the existing CAR T cell technology open here as well. So yes, I can't give you an exact date, but it is moving forward every day and getting closer and closer. And I've had some really exciting conversations with our cell and gene therapy center here, as well as the facilities who actually manufacture the cells. So things are really moving. So very, very exciting.
And I'll just tag on that there are other sites that are going to open soon. I'm here at this conference with Stephen Gottschalk, who runs the immunotherapy division at St. Jude, and they're getting pretty close to opening their own B7H3 CAR T cell trial for both patients with solid tumors and a separate trial for children with brain and spinal cord tumors, and many other sites. I think, now I put it in the chat, but there's 4 sites that I know of that have trials. We have 3 open, and then Baylor, City of Hope, and Stanford each have 1 each. But all of those places plan to have more. City of Hope, someone asked about adults. City of Hope, Mike Jensen, who runs our immunotherapy program in Seattle, was at City of Hope for a while and helped build theirs. They treat hundreds of adults. So children or young adults or actual adults should look to City of Hope if you're looking for adult options. They have a ton of experience and multiple trials open at any given time. And then obviously what our goal is, Hideo Okada's obviously one of the international leaders for a long time in doing this and has exciting work coming through PNOC. And I think on the CAR T cell side, Jess and I have been partnering a lot, trying to get work done in a variety of ways, looking at where's the best place to infuse CAR T cells, which location of the brain, which CAR T cells work the best, which work in the best order, which work in the best group. And our goal is that we're going to be opening multi-site trials. So we have funding now that our next CAR T cell trial in Seattle will be a multi-site trial, and almost definitely, if it doesn't open through PNOC, will include PNOC sites. So, some of these things for regulatory reasons might not fall under PNOC because it's not genetically modified product things that are required, but we'll open the trial at similar sites because we all have shared expertise. So it would be very reasonable to think our trial might open at places like CHOP and UCSF, and we would be working on them together. And I think that's our hope, is that we're going to be opening trials at more sites across the country. Right now, it's very West Coast heavy, which is different from leukemia, which obviously started at CHOP, really. But I hope there'll be more national options soon, and a lot of programs are going to be having trials soon.
I think one of the questions that often comes up, and also here, for international patients, can they enroll in these trials? And I know there are limitations. I mean, there were definitely limitations during the height of the COVID pandemic, and to my knowledge, so every site handles this a little bit differently. So some sites do accept international patients and that's not just to CAR T cell trials. I think that is universal. So some sites do accept international patients and others don't. For example, at UCSF, we do accept international patients. And I think that sometimes varies also currently where a site is and then where the trial is, etc. So I think that is a very trial specific and site specific question that if you're interested, I think you need to reach out to each of the individual sites and investigators what the current status is. So there's no sort of answer for everything. And then another question was, in general with CAR T cell, there's the concern of inflammation. Can you sort of comment on this and what you anticipate and current treatment approaches for that?
Sure, thank you. And Jess, you should be in charge of cutting me off now that I'm talking so much. But So, we've given 255 intracranial doses now, 245, and we haven't seen diffuse inflammation in the brain. So children with leukemia, which is a free-floating tumor in their blood, who get CAR T cells in their blood, unfortunately, actually, some patients can die from diffuse brain swelling. And that, in a simplified version, they get such big inflammation in their blood that it then spreads to their brain and they get diffuse swelling of their brain. We actually always ran our trials as being intracranially dosed because besides being a better location, we also thought there would be less toxicity, and that's what we've seen so far. So out of the 55 patients we've enrolled, only one severe toxicity, and none of them have had diffuse swelling. There are sometimes, we think that the children often get a fever and a headache afterwards, usually need outpatient management. Some of our younger children, like Gavin, was admitted to the hospital earlier in his treatment for the nights after infusion, but now he just goes home the nights of infusion because his parents have become experienced at managing it and things haven't escalated. So we haven't seen a lot of diffuse inflammation. And the one thing I would qualify is, immunotherapy is a gigantic field. CAR T cells are a gigantic field. Every trial of CAR T cells, the CAR is genetically different with different stimulatory domains, which Jess and Jasper explained, that cause different inflammation, cause different receptors to go off. And every one of our trials is different, and they all target different things on the brain, which may or may not also be on the brain tumor, which may or may not be expressed on the brain. So you should not expect side effects to be the same from every CAR T cell trial. That would almost be like saying every trial where a child takes a pill, you should expect the same side effects, because the CAR T cell in some ways is a delivery mechanism to get the T cell to target the tumor. And then for a variety of reasons, you could have very different side effects. So I think we're in a learning phase. Michelle Mange presented virtually at this conference that I'm at, and I think highlighted the experience of her children. We talked about our experience. City of Hope, I'm sure, is different. Baylor is sure is different. And so I expect that all of these trials will be quite different. There will be some overlapping things, but the other difference is some are IV, some are intracranial, some are every week, some are every other week, some are once a month. So we're still at the, unfortunately, the truth is the absolute ground floor of finding out what these can do. And so even though we're learning a lot about the side effects, I think it's going to be a couple more years until we really know uniformly what we can expect when we give these types of treatments.
Thank you, Nick. So we heard a lot about for DIPGs and diffuse midline gliomas. Anything in terms of immune therapy that the panelists find encouraging, exciting for medulloblastoma? Anything you could, is there anything in the works, anything, that is coming up for patients with medulloblastoma and immunotherapy?
Nick, I'm sure can comment because his CAR trials are open to medulloblastoma patients. And so perhaps you should talk first, Nick, but I can say that there are other exciting targets coming out. I can talk about one of them, which is one that I myself and other people at CHOP have worked on, which is going after a target called GPC2. That CAR is moving forward. And hopefully will be open to not just medulloblastoma, but multiple other brain tumor types as well that might express it in the near future. So that's one I'm particularly keen to see opening in the near future. That's again within CARs. Nick, I don't know if you want to comment as well.
No, I think to move away from CAR T cells, I think I just heard Dean Lee at Nationwide talk about NK cells, which is a different immune cell. I think, there's all these different immune cells, and I think Jess highlighted nicely, T cells are one of evolution's best killing mechanisms and can be very targeted, can be manipulated, can be memory and long lasting. There's some of these other people that are just taking NK cells, which is a different group that normally actually fight viruses and other things in the body, but also fight tumors. And that you can take these NK cells, which stands for natural killer cells, and not do any genetic modifications at all. Just get a whole boatload of natural killer cells and give them to a patient. And they've shown really dramatic benefits in some diseases like AML, which is a very difficult type of leukemia to treat. And so I think more and more you'll be seeing NK cell technology. Right now, for not particularly always scientific reasons, there's a lot more CAR T people than there are NK cell people. But I think there's really interesting work going on in the NK cell field. And I think for diseases like medulloblastoma that when they recur, tend to spread to multiple locations in the brain. Getting IV natural killer cells or intracranial could be a really successful strategy, and that's already being worked on through PNOC.
Yeah, and I think you know, a lot of comments what I see in the chat is
Sabine, this is
yeah.
Hideo Okada. Can I
Yeah.
comment? Excuse me, I have to be on transportation, but I've been closely following all these exciting discussions. And also, sorry, Jess, when you tried to have me speak, I had a little bit of a technical issue. I hope you're hearing me okay now.
Yeah.
Yeah, Nick made it. I wanted to follow up Nick's very important comment about NK cells, because, you know, so the current challenge I've been also seeing, like chat comments, questions from patients, you know, the waiting issue, slot issues currently, although many sites are opening CAR T trials, but the inherent challenge is that currently we rely on autologous T cells, which means patients' own cells. So there are a few lines of challenges sometimes. You know, patients are sick, and patients' lymphocytes may have been decreased because of radiation therapy and so forth. And then it inherently takes some time to generate manufactured cells. And then and there's also a slot issue, like for manufacturing laboratories, they may or may not have a slot, particularly these days, a lot of trials going on for multiple disease entities. So there's a strong rationale to develop what we call off-the-shelf cell therapy products, so that patients don't have to wait, and the cells are always available as the manufactured products. So that Nick mentioned about NK cells, and then NK cells are often used as the allogenic. Allogenic means not necessarily patient's own cells, but off-the-shelf products originated from somebody else, but they are okay to use in other patients. Or also there are engineering technologies moving forward to eliminate issues about using somebody else's T cells by getting rid of immunogenic genes and so forth. And even more excitingly, there's an emerging technology derived from Nobel Prize award technology of iPSC pluripotent stem cells from patients, so that we can essentially have an infinite source of the cells. So these are in a rapid and very exciting area of development. So hopefully near future, those things will become available for pediatric patients, so that availability and efficacy and will be improved for patients needing those therapies. I'm just stopping here. Thank you.
Yeah, thank you, Hideo. One of the comments that comes up from repeated, I think, families is obviously there are now a limited set of CAR T cell trials out there, and as a family, how do you make this, like, which is the best one, right? That's always the question. What has the most promise? And I would love to hear from the panelists how they would approach this, or is it basically just too early to say because they're all early phase studies? And, you know, we don't know enough yet to really comment which one is the best one. But there's also quite, and thank you, Nick, you answered some of this in the chat already. These trials are quite different, right? And they're addressing different targets. So depending on the target, one might be better suitable for a specific subtype of tumors versus another. But could you comment in more general terms? Are there really, can you really make an efficacy-based decision at this point, or too early to say?
I would say emphatically too early to say personally. I think unfortunately, we've been living the last 10 years in this boom and bust cycle of very exciting early data presented about the effects of a drug, and then a full study matures and we don't see the results that were reported with what the initial experience was. And so what I tell patients who are looking to enroll in trials is that what people should realize is a phase 1 trial, which is what we're doing, there's actual objectives, and a phase 1 trial by definition, the number one objective is this tolerable, is that safe? So that's the literal objective of running the trial. And then based on how many children you enroll and other statistical considerations, you can also try to look at what degree of benefit it is. But an example I'll give is that I recently talked to a family who lives in the Southeast United States, a little girl who got radiation for her histone mutant pontine diffuse midline glioma, got no other treatment, and now 3+ years is mostly asymptomatic and only starting to show some progression. If she would have enrolled on any of our CAR T-cell trials, she would be the poster child for how successful our trials are, but she's doing just as well on that. So that's why it gets hard to say what any one patient experience. And I will say, I'm thrilled that Gavin is doing so well. He's the sweetest little boy I've ever met, next to my 5-year-old, I should say. But we don't know if the CAR T cells are having a benefit or if he would be doing just as well. Otherwise, it would be unscientific and disingenuous of me to make that claim. So the reason that you finish a phase 1 and run a phase 2 with more patients is that so you can analyze more patients who are on the treatment and you're hoping giving them a benefit, but you can really start to say, okay, this number of patients are doing well in these different scenarios. We can really say compared to historical controls. Because the one-off system of having like poster children that are representative, I think it's great that it draws excitement. It's great that some of these children are doing so well, but it is not a way to determine if a treatment is working or not.
Thank you, Nick. I think that captures it quite well. I think the challenges when we are faced with these questions, and obviously that's a key question for any family, right? When they're deciding on therapy. I mean, one is obviously decision on side effects, but the other is obviously how does it impact the disease. But I think it's too early, and yeah, these single patient examples can be also misleading. So it remains a challenging topic. One question that also was asked in the chat, is there anything known if cannabis, and that's a frequent topic in our patient population and parents. So I think it would be good if there's anything now. I'd love to hear what the panelists have to say that could stimulate NK cells, or if maybe to even make this a broader, discussion. Anything you would advise against or for the use of cannabis in any of these immunotherapy options or trials, or reason not to use it? Jessica, do you want to start maybe?
Yeah, again, I know that there is a little bit of data out there, I believe, that has shown some immunosuppressive effect of cannabis. I have to go back and dig in that literature a bit more, and I honestly haven't looked recently. Maybe there's more to it, but I would imagine that, again, I would err on the side of caution and probably avoid it because of the concern for potential immunosuppression. If you're about to be on an immunotherapy trial, that would be my bias, but I'm curious to hear what the other panelists have to say.
I agree. I think cannabis, is known to be more immunosuppressive than THC, and that's probably the extent of what I know, but I do think that there's the chance that it could limit immune function. I wouldn't expect it to be a lot, and we have allowed patients to take that or similar agents for like nausea control or anxiety while on our trials. We'll never be able to enroll enough children to know whether there's a difference in those patients versus others. So there's a theoretical detriment, but definitely not like a proven benefit or proven detriment that I know of.
Then along the same lines, obviously the question of steroids, does taking steroids before, after radiation therapy, during radiation therapy impact? And I can maybe comment where we looked at this specifically with Dr. Okada in our vaccine trials. Clearly a negative predictor in terms of developing specific immune cells against the H3.3 mutation was the use of steroids. So I think there's definitely, at least in that setting, and that was a vaccine, immunotherapy trial, a negative impact. In general, we think about steroids having a negative impact. The question is really the timing and the recovery of immune system, right? If you were started for 3 days just prior of start of radiation therapy, does that negatively impact you? I don't know if that's necessarily known. However, if you been on it for a longer period of time? My assumption would be yes, but Nick or Jessica or Jasper, any thoughts or comments?
There's really good adult data. Eric Holland at the Fred Hutch has done this both in the lab and looked in adults treated with GBM, but adults with glioblastoma definitively do worse if they're on steroids for prolonged periods of time when you control for everything else. And I think, zooming in on DIPG, I think many of us feel like children who are on steroids for longer, like all the way through radiation and afterwards, do worse. And there's the potential that radiation isn't working as effectively while they're on steroids, obviously knowing that they can be required for some periods. But I think most of us work at the soonest time point we can for that reason, along with to limit side effects. From the immunotherapy perspective, we've only had 2 of our 55 enrolled patients not be able to make CAR T cells from their T cells. One of those was a little kid who had a pretty bad case of COVID, which could have been related. And the other one was on very high dose steroids for a long time. And we weaned low just before they enrolled on the trial, but probably still had some negative effect of steroids. So it wouldn't be a surprise if when you're on treatment, steroids also limit the effect of immunotherapy. Although for like CAR T cells for leukemia, children with leukemia can get CAR T cells, require steroids for a few days and still be cured of their disease. So it's not an absolute limitation to the benefit of immunotherapy, but I think increases the chance that the immunotherapy might not function as well or in the ways that you expect it to.
Sabine, I know
Yes.
you're probably fielding a couple more questions. Let me know
Yeah,
if you.
yeah.
Okay.
So Jasper, that's a question for you. Any news on CAR T cell trials to be open in Europe?
Good question. Of course, we are lagging behind a bit in terms of CAR T development in Europe, but now we are working together with colleagues from the UK to get a trial open in Europe, so the UK and most likely the Netherlands. That's, I mean, we're looking for to get it funded, but we really hope we can open a CAR T trial, but it won't be this year, most likely not next year as well. There are CAR Ts for none, of course, for ALL, but also for neuroblastoma. They're a bit farther ahead, but we definitely hope we can open CAR T trials, say 2023-24 in Europe.
And then maybe I will address this to Ellen and Bruce. There was a comment from a family how a family can have the most impact really on brain tumor research, on pediatric brain tumor research.
Bruce, do you want to start that? And I know you've got some thoughts too.
Yeah, look, this is an area that's just grossly underfunded. And it's underfunded for 2 reasons. One is they're very rare diseases. And all of the work that the panel is doing is probably, I would guess, if not 100%, 98% funded through philanthropy, private philanthropy. So you're dealing with a very rare disease with really very low levels of national awareness. So as patient families, you know, one person can make a huge difference. We've seen that time and time again in some of the work that these panelists are doing, and also with a variety of other diseases that, you know, and one that I can think of is a woman called Kathy Giesti, who has had a huge impact on multiple myeloma, and what's happened with that disease. So it's really about anything you can do to help raise money and help raise awareness and be an advocate for this disease and families that are going through it moves the needle forward. And we've just got to keep doing that until we get the critical mass we need to fund a lot of this work faster. You know, if I'll throw it back to the panel and say, you know, what are your biggest barriers to moving forward faster? If anybody on the panel wants to answer that, you know, what are your 2 biggest barriers to moving forward faster with the knowledge that you have today?
It's, I mean, I will start, but in the end, it's always more personnel, more support, right? I think with more people, we would be faster in moving things along in the lab. We would be faster in translating this into the clinic. We would be faster in opening up clinical trials. It's more manpower to really focus on developing new therapies for especially pediatric brain tumor patient, which I think it's getting more attention, which I think is great to see over the last years, but there still remains a significant lack of funding also from industry. I mean, if you just compare, money that is being put also from the NIH and industry to adult cancers like breast cancer, prostate cancer, lung cancers, where obviously many more patients are being affected. It's really minimal the amount we are receiving for funding, but I think the biggest barrier remains manpower. I mean, that's my take on it, but on every level, in the research setting, in the translational setting, in the clinical trial setting.
And just to qualify, Sabine, is that because I just want to qualify that answer, is that because there's a lack of human talent to aim at this, or is there a lack of funding to recruit the human talent?
I don't think there's lack of talent.
Right.
I might be biased.
I think that was an important point to make. When you say it's a manpower issue, it's not that there's a lack of manpower, there's a lack of funding to support the manpower. Yeah,
I think so. I really think, I think it's such a great cause, right? You can attract talent, and I think it's a very appealing cause for many researchers on the clinical side, on the translational side. I think it's really, it's the funding.
Yeah, and I think that don't underestimate you can't underestimate your personal power to move the needle to raise funds and awareness. I mean, we had an example yesterday where we had 2 high school boys in our community who, for their senior project, decided they were going to do a half Ironman to raise money for pediatric brain cancer. And one of the boys is a childhood lymphoma survivor, and they raised $22,000. They actually did it with a month of training. Yesterday, they competed in a half Ironman, which, for those of you who don't know, it's a 1.2-mile swim, a 56-mile bike, and then a half marathon. They raised $22,000, and they've donated the money to PNOC. So, there's just so much that can be done. And we're here, look, if you like to have some kind of fundraiser, or you need help in actually brainstorming or thinking about how you might be able to do that, we're more than happy to help. You can reach out to Alan, Rachel, or I, and we'd be happy to help you facilitate that. And then on the national level, we're trying to partner with corporations and big brothers and big sisters to really raise the awareness here, and changing the narrative from this is a rare disease to, we frame it in the years of life lost to pediatric cancer every year. When you think about children that we do lose to pediatric brain cancer, they're very young compared to the average life expectancy. And it's really tens of thousands of years of human life that we lose every year to this disease. And that's how we've got to get people thinking about it and the impact that those kids could have on the world if we didn't lose them. So, it's obviously something we're super passionate about. We've just got to continue to get better results in fundraising to support the science.
Well, thank you, Bruce. I think the other comment, what you can have as an impact is really because a lot of our research, right, for some of the tumors, we are so limited in terms of tissue. And so I think, for families to really think about tissue, and I don't know, Eleanor or Rachel, if you brought this up and want to comment on this a little bit more, but I do think that's another important way how families can have an impact overall on the field.
So I can answer a bit of that too, and Rachel could too, because of her work with the ATR registry group. But definitely being a part of with your medical team and making sure that that your particular child's specimen is really available, because as we've all talked about, this is so rare. We just don't have enough of that information. So, and maybe Rachel add that what they're doing too with the ATRT group.
Yeah, for sure. And hello to some of the ATRT families that we've been collaborating on. I see their names popping up. So yeah, I've been working with the ATRT research group with PNOC to basically fundraise $750,000 to fund an ATRT registry. And the idea is that, you know, approximately 50 cases in the year in the US every year, but to have as much data as possible so that just can sort of make the next move. So there is an active call if anybody is interested in. You can always email me if you're interested in participating. It's not open yet, but basically we are kind of collecting any names and emails of interested patient families so that when it does open the registry, that data can kind of be entered in there. So yeah, that's one way to make an impact for sure.
Yeah, and I would just add to that, you know, anybody can make a donation at PNOCfoundation.org to actually support the work of PNOC, as well as any of the other supporting foundations that have participated here today, that you know, are all passionate about changing the course of this disease for their specific tumor types.
Yeah, just on that note too, we want to really thank those particular foundations coming together and sharing with their patient communities, just like Dr. Mueller and all of the other PNOC PIs that are actually collaborating so uniquely and outside of their medical institutions to share information to get this work done, because I don't think it'll really happen otherwise at the pace that we need it to. So I really wanted to just thank them and give them a shout out too. Obviously we have Children's Brain Tumor Network, we have Tough Together, Dragon Master, Tommy Strong, Jack's Angels, Storm the Heavens, we have CERN, NBTS, Hope for ATRT, and the RCDF Foundation. All their communities are joining in and sharing this information to their patient families, so thank you. You know, it does take us all coming together to really make this difference in making sure patient families' needs are getting addressed.
And I want to thank our incredible panelists for the generosity of their time today and the generosity of their knowledge. I mean, it's just such a unique opportunity to have a group of expert panelists like this really talking to us about the cutting edge of the application of immunotherapy to pediatric brain cancers. And, you know, we've done quite a few of these webinars. I shouldn't say I've had a favorite. I've really enjoyed the discussion today, and I'm really inspired and excited about, you know, where immunotherapy could go for patients and patient families in the future. So I'm going to say thank you. Anything else that anybody would like to add before we wrap up?
I'd just like to mention that we are recording this today, and all of our disease-specific previous webinars are in a playlist on our YouTube channel. So everybody will receive an email tomorrow, and feel free to share that with any other patient families that you think might benefit. We definitely want to get the word out to as many people as possible so that everybody has the best information at hand.
Yes, thank you to all.
Everyone. Bye.
Thanks, everybody.
Thanks a lot.
Mhm.
Bye.
Thank you.